Thank you very much. One thing I'm normally asked when I give a talk is what's that when I put it up on the computer screen, and that is Debian Linux, which is what I tend to use on my laptop. So firstly, thank you to Yuval and Graham, and also to Delight for hosting. I'm going to talk about exploring spatial data in R, so this is not a technical talk in any way. In fact, I'm not even an expert in any way in spatial stats, but I notice you haven't had anything in your program yet on spatial data. So what I'm going to do is I'm just going to talk about the general types of spatial data, and then I'm going to introduce the main statistical concepts you need to think about for each of those types. And I'm going to do that by using a collection of examples within R. So I don't really have any slides, a couple. I'll be running them in R while I do them. And so it will be by example. And so you'll learn the main concepts to think about with each of these data types. The question is, what are the data types? Well, no classification is perfect, including this one, but it's a nice classification to have. And this is a statistical classification of different types of spatial data. So the first one is point processes or point patterns. So for example, you might treat trees in a forest, or perhaps cells under a microscope, or perhaps disease cases in an area. You could treat all of those as a as a point pattern. Why might you do that? What's the common link? The common link is you're interested in where the points are in space. Okay. Are there more disease cases to the left of my area or the right of my area? Okay, do the diseases cluster? Those sorts of things. So that's the first type. The second type is geostatistical data. That's actually similar to point process data, and in fact in a data sense it's the same. But with geostatistical data, you have points in space and those points generate data. And you're not interested in modelling the points per se, you're interested in modelling the data that those points generate. A common example is weather stations. Okay, weather stations are points in space. You're not interested in modelling where the weather stations are as such, probably not. You're more interested in modelling the data they collect, wind speeds, temperature, rainfall, etc. Another example is where you drill down into the ground in geology and you get observations on the chemical makeup of soil samples. You're not interested in modelling where you drill. In fact, you may have even chosen where to drill, in which case you're definitely not interested in modelling it. But you're interested in modelling the data that those drilling sites generate in terms of the soil sample. So in terms of the actual data set, the first two are often identical, but there is a statistical division, which is what are you interested in? Are you interested in the points in space, or are you interested in the data that those points generate? The third classification is aerial data, which is you don't have points in space anymore. What you have is areas in space. Postcode regions, census regions, and so on. So I might have counts of autism cases in each Australian postcode. That would be aerial data because the counts don't relate to a particular point. They relate to the entire postcode region. So often the data is aggregated over certain regions, or areas, hence aerial. So aerial data is a different type of data. In fact, the first two types are point data. The third type is often known as polygon data in a mapping sense, because you've got different regions. A mapping classification of data is often points, polygons, and lines. So the first two are point, and the third one's a polygon. Line data is where you have, for example, rivers or roads or geological faults. Okay. So you have lines in space, that's not covered there at all. But often, and I'll show you an example in statistics, where you have line data, you often turn it into a covariate by considering the distance to the closest road or the distance to the closest geological fault. So there's a statistical classification of spatial data. A mapping classification is often points, lines, polygons. Again, this is not the be-all and end-all. You might have data that doesn't fit into any of these types. In particular, I'm not talking about spatial temporal data. So I'm not talking about what happens over time, I'm just talking about what happens over space. So the next question is, what R packages do I use? Well, that's a good question. You can go on the CRAN task view and it will tell you that there's 100 or more on spatial data. Here's the main ones as I see it. Okay, for point process data, you've got StatStat, which is by Adrian Baddeley and Rolf Turner. You've got Splanks is the other one, so they're the main two. Splanks is by Peter Diggle and Barry Rowlandson at Lancaster University, which I was a PhD student at incidentally. So StatStat and Splanks are your main two packages for point data. Your main two packages for geostatistical data is Gstat and GeoR. So Gstat is by a few people, GeoR is by Paulo Ribeiro. The aerial data, the main two are SPDEP by Roger Bivand and D-cluster by a few people. The D in D-cluster stands for disease because often with aerial data you have counts of disease incidents, but it can be used more generally. So I would say there are two main packages in each of those areas. StatStat and Splanks for point, Gstat and GeoR for geostatistical data. You've got SPDEP and D-cluster for aerial data. There's also a recommended package called Spatial, which isn't really much use. It contains a small amount of functions for point data and geostatistical data. A major issue with any spatial analysis is input-output issue. Okay? I want to draw a map of Thailand. Well, you need to get something and get it into R in order to draw that map. Okay, so that's an input issue. How about I do a spatial interpolation? I might want to write that out to a mapping program. Okay. Mapping programs like Google Earth, for example. Quantum GIS is another mapping. So input is a big issue, output is an equally big issue, because you actually need to plot these things. And the main packages for that is SP, RGDAO, and MapTools. Usually, generally speaking, a good idea is just to install all of these things at the start. You don't have to worry about it too much. Because eventually you'll come across something that you don't have if you don't install all of these packages. So they're the main things. So I guess the take home from this talk is that R is excellent at spatial statistics, which of course is what you would think, because R is a statistical program, it should be excellent at spatial statistics. It's okay for static mapping. I'm just going to plot static maps, it's okay. For interactive mapping, it's not so good. Okay? I can't double click on a map to zoom in like I can in Google Earth. It has a small amount of utilities for interactive mapping, but it's quite, it's not designed for it. I'm not criticising R where I say it's bad for interactive mapping. I mean, it's not a mapping program, it's the stats program. So if you want that interactive mapping, what do? Well, you write out your inferences to other programs and do your interactive mapping. So, what I'm going to do, before I actually go through the stat stuff, to show you different maps in R and so on, I'm actually going to show you all the pictures I'm going to use for this talk now. Okay? So I'm just going to run a script. I'm just going to show you all the maps and pictures in R, just so you get an idea of what sort of things you can produce if you wish to. So that's an example of point process data, and obviously I'll explain what these things are in more detail when I actually do the talk. That's a model for the point process data, that's another model, more point process data, some descriptive functions for point process data, some more descriptive functions for point process data, even more point process data with the covariate, another descriptive stats, some models. This is point process data where my points have marks or attributes. This is actually corresponding to cases of leukemia and controls for leukemia data. This is a region in the north of England. In fact, if you're English, you might recognise the lower boundary as the northern bank of the Humber Estuary. And some other things which I'll talk about when I go on. So that's all my point process data pictures. My geostatistical data pictures are here. clearly I've got points in space, but I've now got measurements at each of those points. In this case, I've got zinc amounts in the soil in parts per million, and there's a bubble plot here to represent how large the observation is. Obviously, the larger circles correspond to more zinc, the smaller circles correspond to less zinc. So I'm not going to treat this as point process because I'm not really interested in where the points are as such. I'm actually interested in the data we have at those points. And so this is geostatistical. So I've got a few models for that. Similar data but a different pictorial perspective on it there. I'll talk about this later, which is something called a variogram. And here's some simulations. Okay, so that's the geostatistical example. Second geostatistical example. It takes a bit of time to plot Queensland. I'm not quite sure why. Okay, so eventually Queensland will come up here. My theory on this is that Queensland has so many islands and so many polygons on the edge, Great Barrier Reef. That's why Queensland takes a bit of time to plot. So this again is geostatistical data. These sites are actually the high quality rainfall network sites in Australia as provided by the Met Office. So each of these points represents a rainfall collection point. You might be surprised there's not many of them, but these are the ones specified as, quote, high quality, unquote, by the Australian Met Office. There's only about 300 for them. I can take a look at Victoria, of course. I can't zoom in by double clicking, but I can kind of play about with it and get a map eventually. And here's Victoria, and here's the rainfall collection stations. I do have a small amount of interactivity in R. For example, I can use the identify function, and I can have a look at, say, this point here, which is the rainfall station at Mildura Airport. And there's also one, you might have been here, at Wilson's Prom Lighthouse. Okay. There's a rainfall collection there. So clearly, I've got some interactivity. I can click on my plots, but that's about it with R in terms of interactive graphics. So here's a model. There's another model. I'm not plotting the boundary because it would take a bit of time. Here's another model for my Victorian rainfall. Here's yet another model. Here's something called a variogram, which I'll talk about later. I'll talk about that later as well. Hopefully you recognise this. Do you recognise this? Scotland, yes. I recognise this, obviously. So this is Scotland, so north of Scotland where there isn't much of a high population. And then you've got Edinburgh and Glasgow around here. And as with most aerial data, you tend to get smaller areas. Because you use postcode regions and things like that, the smaller areas tend to be the areas with the larger populations. Where the larger areas tend to have nobody in it at all. So that's a map of Scotland. Here's a few maps of Scotland. Here's a few more maps of Scotland. So that's aerial data, I should have said that. Obviously, when I map of Scotland, I'm onto aerial data because I've got data in each area. And I can use quite vivid colours with aerial data. So that's quite nice. So I've got an observation in each area. In this case, this is something called a standardised mortality ratio for a rare type of cancer, which is basically the ratio between the observed deaths from that cancer divided by the expected number of deaths given the population within that area. So it's a risk measure. It's a risk measure. So I'll talk about that more when I get to that point. Assuming I get to that point. With aerial data, often to do things that correspond to the geography, you often have to define neighbours. So this is my definition for neighbours. This is New York State, Upper New York State, and this is actually leukemia data again. And again, I'll talk about that when I get to it. In fact, this is residuals from a model. So that's that. So I won't finish there. I'll carry on and I'll explain the statistical concepts using those graphs. But, I mean, the main thing is that I think R is quite good at doing these things. Okay. Yeah, it's just a question of actually getting a file, which is often called a shape file, but it depends on the format, to define what object you're looking at and then loading that file into R. So it's fairly easy to draw maps and things in R. It's not that difficult. But of course, I can't, for example, on that Scotland data where I had the Edinburgh and Glasgow, because I've got very small areas there, I would love to zoom in on that. Zoom in on Edinburgh and Glasgow. Okay, zoom in on the city areas so you can see them better. Well, that's non-trivial. You can't just double-click and zoom in. You have to actually subset the object. So you have to have some data which says, you know, these are the areas I want to zoom in on, so you can subset it and then just plot that, like I did with Victoria. Okay. Because the Australian data had a state code in it, so it was quite easy to subset that object, take out the data for Victoria and just plot that. But that was because it had that state code in. If it didn't have that state code in, I would have been in trouble. So it's pretty good for static maps, not so good for interactive. Okay, so let's do all this again, and now I'll talk about the stats behind each one. Before I start talking about the statistical issues, are there any questions? Maps or anything like that?
One question. The files that you need to load in order to generate those maps, do you get them for free?
Do you get them for free? That's a good question. The answer is sometimes. Sometimes. Okay. There are companies that have all these shape files and they charge money, but often you can get them for free. It obviously depends what you want. The Australia data, for example, you can get from ABS, Australian Bureau of Stats website. In fact, from their census, they also distribute shape files corresponding to their census regions. So you can have shape files corresponding to the state boundaries or the local government areas, or even smaller than that, postcode regions. So in Australia, it's fairly easy. But of course, if you wanted, I don't know, Burma or North Korea, or something like that, you might be struggling to find a government website which will give you a shape file for it. I don't know. And of course, the companies that charge for these things say that theirs is more accurate and things like that. So lots of them are available for free. But yeah, if what you want isn't available, that doesn't help you much. So you might have to pay, it might be available. And generally speaking, if you're looking at standard regions in countries like America or Australia or the UK, you can get it for free. Okay, this is point process data. This is maple trees in Michigan, a forest in Michigan. This is observed on a square region. Bear in mind, with point process data, the region of observation is quite important. If you don't have it, you can't really do anything. That's because for all the stat stuff, you don't just need to know where the points are, you also need to know where the points are not. Okay? And if you don't know the region of observation, you can't determine where the points are not. So the region of observation is square here. What are the main concepts I'm looking at with point process data? The first thing is the intensity. Okay, where are most of the points? You've got high intensity where you've got lots of points. You've got low intensity where you don't have that many points. So at the top left, we've got a fairly low intensity. The bottom half of this region, we've got a fairly high intensity. And we can model that intensity. What you typically do is you take the intensity and you put a linear model on that, on the logarithm of the intensity. Because the intensity is positive, so you take logs to transform it to the entire real line. So let me show you a couple of models. The first one is where I'm just going to take x coordinate and the y coordinate. And the reason I'm saying x and y is I'm not actually quite sure what the coordinate reference system is here. It might be latitude and longitude, it might be something else completely different. Okay. So I'm going to call it x and y. So what I can do is I can fit a parametric model to the intensity. I could, for example, fit a quadratic model, which means doing a linear model against x, y, x times y, x squared, and y squared. And a quadratic model, when you fit it to the intensity, that's a quadratic model. It has the kind of quadratic curves in the image plot, as you see there. So this is a fitted quadratic model in x and y, which might be longitude and latitude, I don't know. And you can see I've got a kind of heat map type thing here, and the red area is the higher intensity, and the yellow area is the lower intensity. That's a summary of the intensity of the data across the space. Of course, I can do lots of other things. One other thing I can do is, rather than using parametrics, I can use non-parametrics. And if you've ever done a kernel density estimator, where it's pretty much the same principle here, except you've got 2 dimensions. And so here's a non-parametric model for the intensity. You can see it's a bit more wiggly. The smoothness is defined by the bandwidth. If you have a large bandwidth, it's going to be very smooth. If you have a small bandwidth, it's going to be very variable. Just like with a one-dimensional kernel density estimator. Okay. So I just chose the bandwidth so it looked nice. So you can see it's quite similar to the quadratic with the bandwidth that I chose, but obviously you get the non-parametric shapes of the colours coming out. Okay, here's 3 data sets for you in one go. All on a square region. They've been scaled so that they're all on a unit square. The top left, I've got Californian redwood trees in a forest observed on a square region. The bottom right, I've got Japanese pines, which is another tree observed on a square region. And then the bottom left, I've got biological cells observed under a microscope. Most of the examples I'm giving in this talk are fairly well known, and these are fairly well known as examples to demonstrate the concept of clustering. Okay, what's the difference in these 3? I hope you can see it. Redwood trees have lots of areas where the points are clustered together, so they tend to attract each other. The cells data has the opposite, has inhibition, okay, where one point tends to repel the other point, because it's very regular. And then the Japanese pines are between the 2. So with point process data, you've got 2 issues. You've got the overall intensity across the space, that's the kind of more global issue. And then you've got the more local issue, which is the clustering, or indeed the opposite of clustering, which is here. So in this case, the intensity is pretty much constant over the space, in a global sense. There's no areas with missing points. But you do get different behaviour according to the clustering. Clearly the redwood, you've got some clustering going on. The cells, you've got the opposite, and Japanese, you're between the 2. Often when you look at clustering, you want to decide how different it is from complete spatial randomness. What's complete spatial randomness? Complete spatial randomness is like, complete spatial randomness means that any point is equally likely to be anywhere in the space. So it's like throwing darts at a dartboard in a very random fashion. Okay? They could land anywhere. So if you throw a few darts at a square dartboard, it might look like the Japanese data. The other thing about complete spatial randomness is that the number of points in space are also random. Okay? In a sense, you forget how many darts you've thrown, because the number of points is something called a Poisson distribution, which is where the Poisson process terminology comes from. So when you do look at these things, what you're looking at is really comparing them with complete spatial randomness. How do you compare that? Well, you can use lots of diagnostics. There are lots of functions available based on distances between points. Okay. and the distribution of distances between points in your data, in order to try and identify whether you have clustering, or whether you have inhibition, or whether you're effectively here, which is completely spatially random. There's lots of these functions. There's the F function, the G function, the J function, the K function. There's a normalization of the K function, which I think is called the L function. Okay. So for quite a few letters of the alphabet, you'll find a function. So in SpatStat, there's an easy way of plotting lots of functions for a particular data set. So let's have a look at the functions for it's the cells data. So this is the cells data. So at the top left, I've got the F. Top right, I've got the G. Top left, I've got the J. Bottom right, I've got the K. This is all for the cells data. This is the kind of default image produced by SpatStat. I think it's a bit busy myself. What you're looking at here is really you're comparing two lines. The black line, and the green line, and the red line, are estimates of the function from your data. Okay, so the data-based estimates of the function. So you can pick any of those. I'll consider the black one. So what I'm doing is I'm comparing the black line against the blue line, which is what my function would look like if I didn't have any clustering. In other words, if I was completely spatially random. So what you're doing is you're comparing the blue line with the black line. What's happening with the G and the K is that the blue line is above, the black line is below the blue line. And what's happening with the F and the J is that the black line is above the blue line. Okay? And that's the behavior you get when you have inhibition. Okay, so the opposite of clustering. So this is the cells data where they have a very regular pattern. Okay? So this indicates you have points that are repelling each other. I can look at the same thing for the Japanese pines, and you can see there's no evidence really against complete spatial randomness here. The black line in comparison with the blue line are virtually the same. Okay, so again, the blue line is the theoretical line, and the black line is data-based. So for the Japanese pines data, they're all virtually the same. So there's no evidence against a complete spatial randomness. The third data set goes in the opposite direction, which is the one where we had clustering, which is the redwood data. You see for the F function and the J function, the black line is below the blue line, and for the G function, the black line is above the blue line. For the K function, we would expect the black line to be above the blue line as well, if you have clustering. Interestingly, the K function doesn't seem to pick up the difference, which I think the reason that is, with the redwood data, if you had a closer look at it, the clustering was kind of only in one half of the space. So I think the K function doesn't actually pick up that clustering because the other half of the space didn't have that clustering. So it wasn't really picked up by the K function, often known as Ripley's K function after Brian Ripley, the Oxford academic. So K, G, F and J are really descriptive measures to try and identify clustering. As I say, I'm not really a big fan of how SpatStat does it automatically because I just think there's too many lines in there. I think a much nicer example is from Roger Bivan's book, which presents it like this. And this is for all three data sets using the G function. So this is the same stuff we had before, just presented in a different way, and a nicer way, I think. There's less going on here, and it's easier to pick out what's happening. The black line, and now I've got the G function for all three data in the same plot. The black line is from the data set itself. Okay. The grey area is an envelope for complete spatial randomness. So in other words, if your black line is contained within the grey envelope, your data set, there's no evidence against complete spatial randomness, which is the case with the Japanese pines. But with the cell data, the black line is clearly below that grey envelope. And with the redwood data, the black line is clearly above that grey envelope. So here you're indicating clustering, and here you're indicating regular behaviour. And you can do this with the other functions as well, such as, that's the F function. and that's the K function. Again, you can see it isn't really picking up the difference in the redwood very well, although the black line is still above the simulated envelope. So the other advantage of this is it gives you some idea about the variation, whereas the plot from SpatStat doesn't. It just gives you a blue line. You know, if you have a difference between the black line and the blue line, well, is that a significant difference or not? Well, you can't tell from that plot because it doesn't tell you about variation. Whereas these plots, because of that grey band, tell you about the variation in the theoretical line for complete spatial ranges. So those were a couple of examples. Really just getting to the two main issues you have to think about for point process data. The global intensity across the space and the local issues of clustering. I should say, if you've got both issues going on, you need to model the intensity first before you look at the clustering, and then you look at clustering of what's left over. So I'm essentially assuming here that I've got a constant intensity across the space. Yep.
What would you like me to see if you had both clustering and inhibition in the same?
Yeah, good question. I haven't got an example like that. I probably avoided it deliberately. I think, you know, I've talked about these as two separate issues. Sometimes it's not easy to separate those issues. Okay. It depends on the spatial scale. You know, is that lots of points to the left of my region, or is that a clustering issue? So sometimes it's not kind of easy to separate those two issues. But I think what you do is you fit your intensity first, then what you would do. There's various methods for kind of transforming that and applying variations of these functions to data where you fitted an intensity. So in effect, your fit goes into these functions and it takes account. But the actual result from the images should be the same, in the sense that, you know, if you have clustering above that intensity, clustering beyond what you fitted as an intensity, then you should see the black line above the envelope and so on. But you would use a modification of the K function, inhomogeneous K function.
How do you interpret it for black lines above and below in different places?
I don't know. The function on the x-axis is really about distance between points. So it's tricky. I think it would happen rarely. How would you interpret it? I guess I don't really know is the answer. I think it would depend on the function. You have to look at the mathematics for that. Maybe there's somebody here who's better in a position to answer that than I am. I'm not sure. I haven't really seen it happen much. Yeah, it may be that. Yeah, I have to think about that. You probably have to look at the maths and really think about what that would actually mean. It's not a question I can answer, unfortunately. Any other questions?
Is the region always like just a band, or a region can be any other shape? Because if it's always like a band, just a thickness thicker than the line, then maybe it's like whether the black line is above or below. So
yeah, the region. Well,
the region is more different shape.
Well, the region will always be like a band. It might be like a cone going out because there's more variation at the end here. But it will always be a band because it's based on what's called a simulated confidence. Excuse me, a simulated confidence envelope. So.
So perhaps it's again like above or below, kind of just knowing the data. Above, maybe it's clustered.
Yeah, I mean, it depends on the function. Here, the K function means that if you're above, you're more clustered. So it depends on the function for the above and below. There are lots of functions out there. There's even one sort of modification of the K function that I haven't showed you. So there's a few kind of descriptive stats functions to get some idea of whether you have clustering in your data. What I haven't done here, but what I would say is that you can actually fit models which account for clustering as well. It won't be something called a Poisson process, it will be something else. But there are models available that you can fit to clustered point process. So what other things might you have with point process data? Well, there's a couple of other things you might have. The first thing you might have is covariates. Okay, so covariate, just like in a regression, covariate or predictor or whatever you want to call it, feature. In the spatial sense, the main thing about the covariate is it has to be known across the entire space. So here's an example of a region in Queensland, and each of the points correspond to copper deposits. The line data corresponds to fault lines, geological fault lines. And so what can I actually do with this line data? Well, perhaps the interest is in whether the copper deposits are more likely to be closer to the fault lines. I can use those fault lines to create a covariate across my region. That's the covariate at the bottom. So the covariate is distance to the closest fault line. So the red areas you're further away from the fault line, and the white areas you're on the fault line. And so you can use covariates in your model for the intensity across the space. We don't have a lot of data here, so we don't really have the data to investigate clustering. We're just investigating the global intensity across the space using this covariate. So what can I do? Well, before fitting a model, let's actually do a plot where I've got distance, I've got the covariate on the x-axis, so I've got distance to the fault line on the x-axis, and on the y-axis I have intensity, so the intensity of the points. So that's that plot there, again with a kind of confidence envelope. And what is it telling you? It's almost flat. Okay. We don't actually have that many points above 3. This is the distance to the fault line. So you're interested in the left-hand side of this plot. Okay. If anything, it's actually telling you that as you increase the distance, the intensity of the points increases, which is the opposite to what you would think. Okay. So I think it's fairly flat given the variation. So it doesn't seem to be doing much. If anything, it's actually increasing slightly, which is kind of the opposite of what you might think, because you might think that the copper deposits would be closer. So from that plot, it doesn't seem to be doing much. Another thing that I can do is do a comparison. So I haven't really got huge amounts of data here. What I've done at the bottom is I've just fitted a linear model. So you saw the quadratic model before. This is a linear model because the colours are separated by straight lines. And it just tells me I've got slightly more intensity as I go up and left. Okay. This is without that covariate, so not incorporating the geological faults. At the top I've got with that covariate, which is incorporating the geological faults. And you can see it's slightly different, but not hugely. And in fact, if you actually look at the model and you do a look at the p-value for the covariate, it isn't significant. So you can do hypothesis test of this as well. Just like in a linear regression, you'll do a hypothesis test of the predictor. So in terms of, at least in terms of the distance to the fault line, it doesn't seem to be doing anything in terms of our spatial point quantity. So there's an example of one covariate. You might have lots of covariates across space. The trees data, you might have, say, slope of the ground, for example. You might think that there's some trees that don't grow on ground that's very sloped, something like that. You might have elevation over the space. So you might have a few covariates that you can feed into your model for the intensity of the points. This is some leukemia data for the Humberside region, which is in the north of England. What I've got here, the green points are cases of leukemia. Now this is an epidemiology example. And the problem with epidemiology is that you've got different populations. And so clearly you expect more cases where you've got more people. And in fact, this is the city of Hull, just above the river, the Humber Estuary. So what do you do about that? Well, what you often do is you randomly select controls from birth registers and so forth. And then you can compare the cases against the controls. And so here the green dots are my cases, and the black squares are my controls. Okay? So the green dots are cases of leukemia, the black squares are just ordinary people. Well, not, I shouldn't say that. Normal people without leukemia. So cases from birth registers, and I think these were from birth registers. So what do we do here? Well, you have to obviously, when you look at the cases of leukemia, you have to account for the population. We take the controls. Let's have a look at the intensity of both. Incidentally, this is known as a marked point process, or a point process with attributes, because the point process itself, the data points, contain other data. In this case, whether you're a case or a control. So here's an intensity, just a quadratic intensity measurement for the controls and the cases. And you see they're fairly similar. And what you tend to do is you take a relative risk measure by combining these two things together. Okay. So it's a bit like taking the case and then dividing by the sum of the two. So the relative risk, you can regard it as the probability of having leukemia given the point in space. Okay. Higher values mean the probability of having leukemia is higher. So if we look at that relative risk and see what happens, which one to keep? Here's my relative risk. And you see it doesn't have much variation until we get to the left-hand side. What this is saying is that the risk of getting leukemia is increasing as we go west. But you've got to be a bit careful with these things. Because bear in mind, with any maps you look at, all you're looking at is a point estimate. Okay? I mean, have a look at why the relative risk is increasing. That's because I've got 4 cases, 1, 2, 3, 4, and I've only got 1 control. So I'm making an assessment based on 5 people. It probably isn't a good idea. Okay? So clearly I've got lots and lots of variation where I don't have data. So with maps, you've really got to be careful that you do understand not only that you've got a point estimate, but that your variation of that estimate changes over the space. So here I might get quite good estimates because I've got lots of data, but here I might get quite bad estimates.
Isn't that the result of you taking the population into consideration when you take control? You sample the control data?
I guess it is in a sense, but what's the alternative? I mean, you have to take the population into consideration. Because what you want is an estimate of the probability of having leukemia given you're in the space. Clearly, if you just look at the leukemia, where the leukemia cases are, well, clearly all of them are going to be in the cities because you've got more people in the cities. So, I mean, you could do an analysis of the data and say, well, there's more leukemia cases in Hull than there is over here, but so what? It's like saying there's more leukemia cases in Melbourne than Simpson Desert. Well, yes, I know that. So you need to take the population into account. So I guess yes is the answer, but you have to do that. So these are my zinc measurements, and so I'm on from point process data to geostatistical data. These are measurements of zinc in soil samples in parts per million. What do I do with this data? Well, again, in point process data, I'm interested in the global intensity. It's a similar thing here, except I have to do spatial interpolation. What does that mean? It means I have to sort of, by fitting a model across the space, what I'm doing is I want to estimate the concentration of zinc at points where I don't have data. So I want to estimate the concentration of zinc across the entire space, not just where I actually have the data points. And that's a spatial interpolation issue. What do I do with that? Well, I can do a few things. I can actually just use a weighted average based on Euclidean distance. So let's see what a weighted average gives me. That's what a weighted average gives me. Not very satisfactory. because I end up with these sort of peaks and troughs where I have my points. So I have that big point there, and I have this kind of red peak. And so it's not very smooth and not very satisfactory. But that's literally just a weighted average. So at each point, I just take a weighted average of the data around it, okay, based on the distance to that data. So that's something simple I can do, but clearly it's not very satisfactory interpolation. Instead of using a weighted average, let's say I couldn't use linear regression. Straightforward, linear regression. I could take latitudes and longitudes and do a quadratic model, a bit like I did with the intensity before. That would give me something like that, which seems a bit more sensible. We're kind of getting somewhere. Obviously, it's a lot smoother. Then I could be a bit more clever about this and think, well, do I really want to use latitude and longitude? Could I use some other thing? And I could see there that I've got high zinc deposits near my river. So instead of latitude and longitude, let's use the distance to the river. So this here, I should say, sorry if I said this, this is the River Meuse, so the river is the grey strip, and the data is to the east of the river. So let's use the distance to the river as a regression. So I've got one predictor and its distance to the river. And I can do that. And that's just a linear model using distance from the river.
You mean the shortest distance?
Yes, shortest distance, sorry. So shortest distance to the riverbank. And so you see, as I get further and further away from the riverbank, I tend to get less and less predictions for the concentration of zinc. There's no reason I have to do these two things separately. I could put in latitude and longitude, a quadratic model for latitude and longitude, and I can put in shortest distance to the river. I could do that. I get something slightly more satisfactory. So that's fine, but that's still just a linear regression. What's the problem with this? Well, there might be a problem, and there usually is. So let's have a look at the problem. Here's the problem. I've done my linear regression, I've done a quadratic model, latitude, longitude, I've also put in distance to the river, closest distance to the river. So that's great, I've done my linear model, I've done a spatial interpolation based on my linear model. So I've got my fitted values on the left-hand side, I've got my residuals on the right-hand side. This isn't a bubble plot, it uses colours rather than size. So here my red values are large residuals, and my smaller residuals as I go lighter and lighter. Now the problem is a linear model should have independent residuals, right? Once you've fitted your linear model, you should have independent residuals. Well, the same thing applies in space. These residuals should be independent if my model was good. And the problem is they're not. You can see I get groups of kind of red-orangey points in my residuals, kind of grouped together, and that's spatial correlation. So what I've got is I've got spatial correlation. I've got large residuals sort of in groups. And so I've got correlation across space, which I haven't yet accounted for just by doing my linear model. That's what spatial correlation is. So how do I deal with spatial correlation? Well, let's have a think about this. Well, let's suppose I take my residuals. Okay, so bear with me on this one. What I do is I take every pair of points, okay, I plot the squared difference in the zinc concentrations on the x-axis. On the y-axis, so the squared difference in the zinc concentrations on the y-axis, and I plot the distance between the points on the x-axis. This is of my residuals. And that's what I get up the top. Okay, so every point corresponds to a pair of residuals. So here I get the square difference in my residuals. Okay, and on the x-axis I actually get the distance between those 2 points in space. Okay? So what happens is if I smooth these all points out, I get something called a variogram. Okay. So that's my variogram at the bottom. What's the point of this variogram thing? The the point is as follows. If I have 2 points that are close to each other, okay, I might expect my zinc concentrations to be quite similar, okay, because they're close in space. So if they're quite similar, the residuals will be quite similar. And the squared difference will be quite small. But if the two sites have a large distance between them, I might expect the residuals to be quite different. Okay. And therefore the squared difference will be large. So if I have spatial correlation, I'll start small and I'll get larger. Okay. If I don't have any spatial correlation, I'll be flat. This is telling me I have spatial correlation because I start small and get larger. You're not interested in all the distances, so I'm only probably interested in up to about halfway along here. For those who know about these things, the value that it starts at is called the nugget. So the nugget here is about 0.1. So this is the sample variogram. You can think of this as kind of describing the correlation, the spatial correlation of my residuals after I fit my linear model. So the question is, can I use that spatial correlation in any way? And the answer is that you can use it. How might you use it? Well, if you want to use it, firstly, you've got to model it. And I've just put a smooth curve on this. Typically, you'd use a parametric model, but you can use a smooth curve as well. Sometimes it's quite difficult to fit. In this case, here's my model, here's my points, smooth points. This is something known as a spherical model, which has a nugget at about 0.08. So it starts from 0.08. It goes up, and then it flattens off. You wouldn't I know the points go back down, but you wouldn't expect a variogram to go back down. That might just be a consequence of the large variation at large distances. Because at large distances, you don't have that many pairs of points. So clearly, your variation's going up. So this is a parametric model for my spatial correlation. How do I use this? Well, what I do is I can kind of combine the linear modelling stuff, where I model my trend, with the variogram, which is kind of taking into account my spatial correlation. So I can combine these two things together, and how do I do that? I do that using something called kriging, whatever you want to call it. And so the idea of this is that you combine your trend with information about your spatial correlation to get a good interpolation across your region. Okay. So you're using those two aspects of the data. In order to do it, of course, you need to fit a parametric variogram model. Okay. I've done this in kind of 2 stages, but in practice, just for demonstration purposes, in practice you don't do this in 2 stages. It all happens simultaneously. Okay. So you stick in your trend term, you put in your parametric model for your variogram, and the Krige function in R sort of does it all for you. So there's the more realistic interpolation of zinc measurements. And there's simulations. One of the other things about geostatistics is that, again, your surface is a point estimate, but you can simulate to try and get an idea of the variation. So that's one way of doing it, to simulate the surface of the zinc concentrations across the space. Okay, this is my Australian map with my sites. I can't move until it stops plotting, sorry about that. So what I've got is a rainfall network of sites, 350 sites, a high quality rainfall network in Australia. So this is what I'm going to show you in R. I'm actually going to show you it So I'll show you it afterwards. Here's a Victorian map of Victoria. I think I've lost my mouse. Oh no, hang on. Yeah. My mouse has gone. Not sure why. Sorry about that. So here's my Victorian data. What I want to show you is the sites in Google Earth. So if you wait a second, it will fly into the screen. So here's my high quality rainfall network sites. That's just telling me I've got no internet. I've got no internet connection, which is probably why it comes up as crosses. But if I had an internet connection, it would come up as pins. So here's my sites across the region in Google Earth. And I did that by writing out the file that Google Earth uses from R. So there's my Victorian data set. This is again a spatial average. This is a model of the rainfall using quadratic regression. And you can see you tend to get higher rainfall in the tropics and in the east, and in the desert regions, modelling lower rainfall. Again, here's my weighted average model for rainfall across Victoria, and you can see it doesn't do very well. Okay, you've got a very high peak at that one site, which isn't very good because it's not smooth enough. And again, I could do a quadratic model, which is just telling me that I have higher rainfall towards the east. Here's my variograms. That one's for Victoria, so it does seem to be going up a bit. There seems to be some amount of spatial correlation there. And here's for the whole of Australia, just using distances up to 300 kilometres. And again, there seems to be some spatial correlation there as well. So if you wanted to, you could do parametric models for those and incorporate them back into the spatial interpolation. So what I can also do so I can actually load my interpolations into Google Earth like that. Often the images don't tend to work too well, but if I have that interpolation image, I can use my output programs, get that image and put it on Google Earth like that. So still using Google Earth. Let me look at some data from Scotland. Again, I've got no internet, so I'm not quite sure how this is going to look. That seems all right. So here's my Scottish data put in Google Earth. So here's all the regions I have. Here's all the different regions I have. Here's the boundaries of those regions. So that's the data you saw in R.